An inducible operon regulatory gene regulates the mRNA production in which RNA polymerase makes RNA, and this is the regulatory gene. Then the promoter starts the transcription, which is next to the regulatory gene, and the operator activates transcription. The repressor then blocks or restrains the promoters. The inducer binds on the repressor, resulting in an inactive repressor, and changes the shape of the repressor. It then prevents binding to DNA and allows for transcription and gene expression to occur. Types of regulatory molecules that can affect the expression of operons: repressors, activators, and inducers. Regulatory proteins often bind to small molecules, which can make the protein inactive or active by changing the ability to bind DNA. This is the inducible operon.
In the repressible operon, the regulatory gene controls gene expression and limits mRNA production. The RNA polymerase creates mRNA, while the promoter starts transcription, whereas the operator can start and also stop transcription. The repressor blocks the promoter, discontinuing the production of protein. In the repressible operon, it usually starts on, making mRNA, but can be repressed or inhibited to stop, which is the effect of co-repressors on the gene expression.
Eukaryotic gene regulation, which is a regulation of eukaryotic gene expression by a combination of molecules before, during, and after gene transcription and mRNA translation. An intron is a segment of DNA or RNA molecule which doesn't code for proteins and interrupts the sequence of genes. An exon is a segment of the DNA or RNA molecules containing information which codes for protein or peptide sequences. The difference between these two is that an exon is termed as a nucleic acid sequence, which is represented in the RNA molecule, but introns are represented as nucleotide sequences seen within the genes, which are removed through RNA splicing for generating a mature RNA model. Poly-A tails are a long line of adenine nucleotides, which are added to the messenger RNA to stabilize the molecule. 5' cap protects mRNA from degradation during the translation process. So basically, in the nucleus, the chromatin is remodeled at first, then it is transcribed. After it is transcribed, it's open DNA, which then processes the RNA, where the 5' cap and the poly-A tail are used to stabilize the nucleotides. Then after the mRNA strand is stabilized, then it goes to the process of translation, which is where it creates a post-transitional modification, which is just folding the glycosylation transport, and it activates or degrades the protein.